to complete for today. So you're looking for tasks to complete for U2L6. You can find that under assignments or on the Disease and Health homepage in the weekly calendar. Today's date is the 19th of October. Today I want to continue to explore COVID-19 a little bit, and if we have time, we'll do some stuff on the flu. We probably won't, though, save flu for our next influenza for our next lesson. But my goal, just to wrap up this unit, you can kind of see it on the calendar, but I want to dive into a couple specific diseases, specifically flu, rabies, Ebola. There's two other ones. And then we'll finally have our test probably right before Parents' Weekend, just to give you a heads up on that. Okay, as usual, I'll throw you into breakout rooms for a quick little chat about some silly question. Today's question is, you have your own late night talk show. Who do you invite as your first guest? Which is basically an elaborate way of saying, what's a celebrity you would like to have a conversation with? Give you 20 minutes to think about it, and I'll throw you in breakout rooms to share your answers. All right. The next thing I wanted to do was, bear with me, it won't be the most exciting thing, but I found a New York Times article that I thought was really fascinating about coronavirus unveiled. And I just wanted to read two portions of this aloud with you as I share my screen, and I'll show you some of the cool animations. But this came out on October 9th, and you can click on it. As far as I know, you should be able to open the New York Times. If you can't, I have a little shortened PDF version of it right there. But it's all about the chemical composition of this virus. And they have some awesome images. Hopefully that's what you're seeing on your screen right now. And so just to get through this quickly. So in February, as the new coronavirus swept across China, shut down entire cities, a scientist named, I'm going to say Sai Li, set out to paint its portrait. At the time, the best pictures anyone had managed to take were low-resolution images in which the virus looked like a barely discernible smudge. Dr. Li, a structural biologist at, I think it's Tsinghua University in Beijing, joined forces with virologists who were rearing the virus in a biosafety lab in the city of Hangzhou. Those researchers doused the viruses with chemicals to render them harmless and then sent them to Dr. Li. Dr. Li and his colleagues then concentrated the virus-laden fluid from a quart down to a single drop. He could only hope that they had done everything just right so that the weeks of work to produce the drop could not have been a waste. Can you imagine like spilling that? That would have been crazy. At the time, you don't know what's inside, Dr. Li said. It's just liquid, right? Glimpsing the structure, Dr. Li carefully froze the drop in a fraction of a second. If it had the slightest mistake, ice crystals would spear the virus, tearing them apart. Hoping for the best, Dr. Li placed the smidgen of ice into a cryo-electron microscope. The device fired beams of electrons at the sample. As they bounced off the atom inside, Dr. Li's computer reconstructed what the microscope had seen. When the picture formed, it was taken aback. I saw a screen full of viruses, Dr. Li recalled. And so you can see them all here, and then here's a computer-colored rendition. And again, pretty obvious that it's just got a simple sac. It's an RNA virus. See that helical shape? Only RNA viruses can have that helical shape. And it's got these proteins around the outside. That's what it uses to bind to and infect other cells, and they'll go into detail about that. You could see thousands of coronaviruses packed in the ice like jelly beans in a jar. They're beautifully intact, allowing him to inspect details of the virus that measured less than a millionth of an inch. I thought I was the first guy in the world to see the virus in such good resolution, Dr. Li recalled. Over the following weeks, Dr. Li and his colleagues pored over the viruses. They inspected the proteins that studded the surface, and they dove into its core, where the virus's strand of genes has coiled up with proteins. Pictures reminded Dr. Li of eggs in a nest. Kind of a more three-dimensional rendering of that image from earlier. Thanks to the work of the scientists like Dr. Li, the new coronavirus known as SARS-CoV-2 is no longer a cipher. They have come to know it in intimate atomic detail. They've discovered how it uses some of its proteins to slip into cells and how its intimately twisted genes commandeer our biochemistry. They've observed how some viral proteins throw wrenches into our cellular factories, while others build nurseries for making new viruses. And some researchers are using supercomputers to create complete virtual viruses that they hope to use to understand how the real viruses have spread with such devastating ease. This time is unlike anything any of us has experienced, just in terms of the bombardment of data, says Rami Amaro, a computational biologist in the University of California at San Diego. Probing the spike. Earlier this year, Dr. Amaro and other researchers directed much of their attention to the proteins called spikes that stud the virus's surface. Spike proteins have an essential job to play. They latch onto cells in our airway so the virus can slip inside. But it soon became clear that the name is a misnomer. The spike protein is not sharp, narrow, or rigid. Each spike protein snaps together with two others, forming a structure that has a tulip-like shape. A long stem anchors the protein to the virus, and their top looks like a three-part flower. Look at that crazy animation. They're all in motion. Gerard Hummer, a computational biophysicist at Max Planck Institute in Biophysics, and his colleague used the frozen microscopy method to take pictures of spike proteins embedded in the virus membrane. Then they calculated how the atoms of the proteins pushed and pulled on each other. The results have a molecular dance. So here you have the virus membrane on the very outside, and here are these spike proteins which bind to the cells. And it's important to understand these because this is what the virus uses to infect the cells in our respiratory system. You can see these flowers waving with all kinds of bending angles, Dr. Hummer said. It's quite surprising to have such a long, slender stalk with so much flexibility. A sugar shield. Dr. Hummer speculated that the flexibility of the spike was important for the virus's success. By sweeping around, the spike increases its odds of encountering the protein on the surface of our cells it uses to attach. As they sweep around, however, the spikes can be attacked by antibodies, powerful soldiers in our immune system. To hide, they create a shield out of sugar. Sugar molecules in navy blue below swirl around the protein and hide it from the antibodies. I think this is fascinating. So this is the hook that attaches to our cells and allows the virus to infect them. And yet it's sort of incognito. It's almost wearing a disguise of these very complex sugars that I assume our immune system sees as innocuous and doesn't immediately attack them. Anyway, there's a lot more detail about the hooks and looking at how the loops are constructed. And if you want, you can take some time to read through this on your own. I'm just going to skip down to the very end where they then talk about the potential for a vaccine or treatment now that they are understanding more about how coronavirus infects us. Right here. So, drugs and vaccines. Already, however, the new pictures of the SARS-CoV-2 have become essential for the fight against the pandemic. Vaccine developers study the virus's structure to ensure the antibodies made by vaccines grip tightly to the virus. Drug developers are concocting molecules that disrupt the virus by slipping into the nooks and crannies of proteins and jamming their machinery. So it's all about creating proteins that can kind of clog up these spike proteins so they can't hook onto our cells anymore. The virus's genome may offer other targets. Drugs may be able to lock onto loops and tangles and prevent the virus from controlling our ribosomes. It's very important that you know what the shape is so that you can develop the right chemistry to bind to that shape, Dr. Ruskin said. Dr. Gladfelter, meanwhile, wants to see if the physics of viral droplets may offer a new line of attack against SARS-CoV-2. You could get a compound that would make them stickier, make them more jelly, he said. There's probably a lot of Achilles heels. Future research. While the past few months have delivered a flood of data about the virus, some studies have made it clear that it will take years to make sense of SARS-CoV-2. Noam Stern-Ginosar and her colleagues in the Weizmann Institute in Israel, for example, have found evidence that the virus makes proteins that scientists have yet to find. Dr. Stern-Ginosar and her colleagues surveyed the RNA of the virus in infected cells, tallying up all the ribosomes that were reading it. Some ribosomes clustered along known genes, but others were reading genes that had never been found before. Ribosomes sometimes read just a section of the spike protein gene, for example. Presumably they make a mini spike, which may very well carry out some essential job for the virus. A drug that disables it might cure COVID-19. But scientists can't even begin to guess at these possibilities because no one has yet spotted the mini spike in the wild. And the same will be true for the other new genes Dr. Stern-Ginosar's team has found. Each one will require additional work to figure out, and they're doing what they're doing. She said, biology takes time. And I encourage you to check out the middle of that. They go into a lot of detail if you're curious about what's going on with COVID and how it infects things. But anyway, I just found that was really interesting that we've learned a lot about its internal and external structure, and it's kind of raising some hopes about potential vaccines and how they could work, but it's also going to be incredibly complicated. Any questions or comments on that? Okay. The next activity I want to do then is have you reflect on your current local coronavirus situation. This is step number 4 on the tasks list. If you click on the assignment right there, it says do some research and complete the Your Local COVID-19 Situation classwork assignment. I'll share my screen here again really quickly just to walk you through one or two things, and I'll let you go to work on it. But here it's just simple instructions. I would like you all to open this Google Sheet and fill it in using these different research sites. And it looks like some of you are getting going on that. I have mine filled out on the top. I just chose Maine as a random spot. But there's some good research websites here. The data, as the article said in New York Times, is overwhelming. There's so much of it. Two that I'll show you really quickly, though. I think Google is probably the best place to start. If you just Google search global cases of COVID-19, if your computer knows where you are, like mine automatically pulls up Monterey County. But there's a bunch of interesting tools here. You can look at the daily change. You can look at cases, even in your local area. You can change the country. And so you can get down to the county level here if you wanted to. If you're living in the US, USA Facts has a great site. I have a link to it right there. But they have every single county in our country. I wouldn't start here though, actually. I would scroll down to where the states are. You can see this here. So then choose the state you're in. So like this is where I was when I chose Maine. Let's say if you click on California, and then it'll bring up a county map of that state, which is a little more navigable. And you can see Monterey County right there or wherever else you're at. And once you click on that, you can see all the info that is required for the graph. You've got known cases, known deaths, shows you a trend, and you'll just have to kind of add up the number of deaths over the last month. That might be a little tedious, but you might be able to find that information somewhere else as well. Once you have your data filled in on this Google Sheet, I'd like you to fill out this survey. It's an anonymous COVID-19 personal reflection where you'll be reflecting on your data, but also just your experiences over these last few months. I'll time us for about 20 minutes to get through this, but I'll keep an eye on the forms and on the Excel sheet. And once it looks like everybody's done, we'll come back together. Let me know if you have questions or if you're having trouble finding certain information or if you're not sure what to do. Go for it. All right, we are all back. While you were all chatting, I was looking through the responses to the questions, and as expected, most of you are kind of, in terms of personal health, worried about either older family members or yourselves in some ways, and that's definitely been kind of the trend. And good that you're kind of keeping those people in mind. In terms of economic impact, it seems to be across the board, but yeah, same thing, just kind of holding on to jobs or people who are working in certain markets that are having a harder time. One thing that you can do to support, again, I like that a lot of you are aware of, it's tricky, but wearing a mask, staying away from people sometimes, even though you want to go visit, donating food, those are all good things you can do. And these are things I hope that you are doing or that you get inspiration to do. What sucked the most, again, not surprising, and I agree with all this, just kind of missing out on certain experiences. I really miss public pools, and I really miss going to the aquarium. Impact on mental health, they're finding a lot of that now with teenagers in particular, but there's ways to kind of deal with it. And that's why I wanted to end with that last question of just what's been on the bright side, and looks like you're focused more on schooling and spending time with family, learning new skills, being at home. So I encourage you to kind of keep focusing on those things and help us get through this. Again, I'm already very, very impressed with a lot of you and your resiliency throughout all this. Any other questions or comments on that, the reflection? All right, well, in our last few minutes here, I just wanted to move on to one last new disease, move on from COVID and focus on another respiratory disease, which you'll be reading about tonight. I'll just do kind of a basic intro, and once I'm out of time, I'll cut us off. We're going to talk about the flu. because we're about to enter flu season. How many of you have had the flu before? I should be seeing every hand raised. Sophia, you've never had the flu? Really? Oh, all right.
I
Interesting.
also haven't had it before.
Tammy, Bella? Oh, all right. Interesting. Usually, most people, especially by the time your age, you usually had it at least once, but that's great. Hopefully, that continues to be that way. That doesn't spread around anymore. So here we go. You'll be reading Gladwin chapter 24, which is going to go into a lot of detail about the flu. And I'll emphasize, like I did last time, don't get too lost in the details. I know there's a lot of vocab that the reading throws at you sometimes, but just kind of come away with the basics. So, like you showed in our little survey, a lot of you have had the flu before. It comes every season. Some people kind of don't think much of it, but it really is serious. About 20% of the world gets it every year. And we can often have epidemic influenza in the United States. We're about to enter flu season. It's usually between December and March. 10 to 40% of people who are within a community, if it starts to spread, they will infect it, and that includes things like schools. And so that's why you've got to get your flu shot every year. If you have not gotten it yet, I encourage you to go out and get it. I think Stevenson is also requiring it as part of our policy for returning back to campus in January, hopefully. There's two big families that the flu fits into, and I like the way the book kind of breaks it down, that you've got your orthomyoviruses, which are like the ordinary, that's the ordinary flu, and then the paramyo, which is like all the crazy stuff. It's a parade of viruses. That's measles, mumps, some of the more scary things. Flu epidemics always follow the same pattern. It's kind of interesting that usually it's schools or some other community is where it starts, with kids getting sick, and then they start to miss school. Then parents and other adults start to miss work because they're also suddenly getting sick. Hospital admissions then usually rise with pneumonia in particular, which is just lung infections in general, which can be triggered by having the flu, and then some bacterial infection also takes advantage of it and gets in there, or just your lungs are filling up with fluid. And then death does occur usually within the elderly, or in people who are immunocompromised, meaning that they have weakened immune systems. A lot of people confuse the flu with the cold, but there's a couple key distinctions. You're going to have a whole lot of fatigue and weakness, very high fever. That seems to be kind of a really big giveaway, especially in children, and lots of muscle aches. This is kind of the thing, and you often get shivers and stuff as well when you've contracted it. It is an RNA virus and negative strand. If you remember from our earlier virus chapter, that means that it can't infect cells immediately. It has to have its own little RNA polymerase to transcribe it, and then it can go be available to the ribosomes. It is in helical form, just like the coronavirus, which is another kind of indicator that's an RNA virus. And the key thing to focus on is these spike proteins. Same thing that the article about coronavirus is talking about. There's these two different surface proteins which are used to infect your upper respiratory cells. And this is important. You have the hemagglutinin, and the activity of that, that's the infection protein. That's what binds to host cells in your upper respiratory system and then has the transfer of RNA and infects the cell. But it's backed up by what's called neuraminidase, which is essentially cutting past all the defenses that your upper respiratory cells have. You actually have this sort of mucus, it's called mucin. It's a barrier that covers all your upper respiratory cells and make sure that they're not in direct contact with some of the pathogens you're breathing in. And the flu virus essentially has this protein that cuts through that and then allows this protein to then bind to the surface of your upper respiratory cells and infect them. And so it's sort of a two-part thing. And that's going to become important in how we name them. Your book has this kind of goofy image of how they work together, but again, here's the, we'll abbreviate them HA and NA for simplicity, but here's the one that cuts through that defensive barrier, right? So there's no more defenses in your cells. And then this is the infection one, which binds to, it's a celiac acid on the surface of your cells that allows the virus to then infect. So hold up. We know all this, and there are drugs that we have come up with which interfere with the function of these proteins. We can knock out these proteins. Then why are we still having annual flu outbreaks every single year? And it's because the flu virus is constantly changing. It can either change its actual HA and NA proteins, or it can literally exchange and swap them to make new combinations. And this is what's made the flu such a hard thing to vaccinate against in the long term. So there's two things that occur. There's antigenic drift and antigenic shift. And I know that's kind of annoyingly similar descriptions, but you're just going to have to try to make this distinction as best you can. Think of drift as the lighter form, right? This is just a simple mutation in the genes that code for those spike proteins, and the protein just changes its structure a little bit. Your book uses this kind of goofy diagram of like a ship at anchor, and you just move it over a little bit, right? So it's still the same ship, it's still in the same water, it's just a slight change. Antigenic shift is really big, right? It's a giant change. That's why I think this is supposed to be a picture of a giant holding the boat out of water. It's completely different. You have two different flu viruses which essentially exchange these proteins, right? They exchange an HA or an NA, and they form a new combination, often one that has never been seen before. And so the human immune system is totally unprepared for it. And this often occurs inside animals, which is why you get these references to things like swine flu and bird flu, because that's where this is occurring. And I think this is fascinating. So here's a little more detailed diagram. It's the same slide, but again, you can see here's the antigenic shift. This is the more dramatic change, right? Where you've got two different viruses that essentially decide, hey, let's swap our HAs or our NAs. And you get this new formed virus, which is a combination usually of like a human infection virus and an animal infection virus, right? So for example, again, with birds, you have a virus that normally infects humans, pops into a duck, joins up with the flu virus that would normally infect ducks. They swap pieces, and now you have a completely new flu virus that jumps back into humans. And again, this is why we can't vaccinate. Every year there's some kind of new combination, and sometimes every once in a while you get one that is very, very virulent and lethal. And this is just showing drift, where you just have a slight change. You can see that they change the structure from like triangular to round on that one protein. So viruses are named after these proteins. If you've heard of like swine flu, H1N1, right? The H and the N refer to those proteins that I was just talking about that are used for infection. It's just the protein type, so it's just that combination. And hence you can see that's why we have these different combinations of flu virus that are constantly reinventing themselves, basically. But H1N1 is the big one. It caused Spanish flu, which ironically actually did not come from Spain. It actually originated in Kansas, here in the United States, on a military base. But it killed 20 to 50 million people worldwide. This was a really, really bad outbreak in 1918, one of the worst viral outbreaks. And it has had a couple more outbreaks since then, but nothing as severe. 2009 was the most recent one, and that's when it was referred to as swine flu. Not sure why the name changed. I'm sure there's a reason, but and there's been a couple other versions of the flu that have reached epidemic proportions, right? This is showing you the epidemic ones that have become epidemics. And your book goes into detail about aspirin use and things like that, but I will let you read that on your own and cover that for homework. Any questions or comments on that so far about the flu? All right, we'll pick up with our review the next time we meet. Homework is on the bottom of the tasks to complete list. It is to read just 4 pages of Gladwin chapter 24. Please only read page 240 to 244. I have provided a scan as well if you don't have a copy of the textbook. As usual, submit one photo of 1 page of your notes, and there's a little reading reflection to complete, which you can take up to 10 times. Any questions on the homework? All right, that's it for me, kids. Thank you very much. Have a good day.
Thank you.
Thank you, Mr. A.
Thank you.